of synthetic biology and microbiome research. So before we go further, I just wanted to say that what is microbiome? It is very important to understand what is microbiome. So microbiome is nothing but it is the environment in which different types of microorganisms called as microbiome. So about this synthetic biology, this microbiome is also related to the human body. So what is human microbiota? Human microbiota consists of microorganisms including bacteria, viruses, and fungi that live inside the body or on the human body. So these microbial communities are dynamic, means they changes and they keep on changing, and the members of the community fluctuate over time. So if you see the picture or the figure which is given on the right hand side top A, they have shown you that where all we can find the microbiota in human body, for example, skin, in so many other parts. So these microorganisms are already living on us or inside us. So understanding and controlling these microbial communities can help us to maintain the health and treat the disease. And this can be learned only when we know the complex interactions between the microbe and the host, as well as the microbe and the microbe interaction. So these things are studied under system biology. This system biology approaches the aim to describe the complex cellular or tissue interactions by implementing biological networks and using mathematical models. Integration of our current knowledge of system biology methods with the field of synthetic biology provides us an approach to manipulate and design the microbial systems in which the human health and the treatment of the disease can be improved. So, with the help of synthetic biology, we can do all these things. But what is synthetic biology? It is the field that uses engineering strategies, including the models for biological investigation. This can be done by genetic tools by creating synthetic microbial communities, those where species are developed or modified and introduced in the human microbe. For instance, the bacterial stains can be designed to improve a certain pathway, leading to increased production or consumption of certain metabolites, leading to a specific function. This can act as a delivery system at a specific microbial community in the human microbe. So even through research, it has been proved that these synthetic biological organisms can be useful. And this can be done by different methods like metagenomics, then amplicon sequencing, then metaproteomics, metabolomics, and single cell omics. But also it has so many advantages as well as the limitations. This synthetic biology can be applied in various fields, for example, in medicine, agriculture, as well as in industry. In medicine, we synthetic vaccines, use them to get genetically modified crops and to get synthetic fertilizers. In environment, we can use them for bioremediation as well as to get good quality biofuel. Getting new materials. Although it is very useful, but organisms or synthetic biological organisms is very costly as it requires a lab work. Then the promise associated with the synthetic biology does come with the risk also, as it has to identify, mitigate, and to be managed. Regulatory frameworks need to manage the risk and ensure an appropriate balance between the industry, new technologies, and any concern with more broadly. Although it has so so many challenges, but still in future we can work, make great advances in patient care. The option we have is then we can also be able to understand the microbial dynamic in human system, and we can also work on the systems microbiome medicine approaches which are newly or developing future for all of us. These are my references. Thank you.
All right, thank you. Do you have any question for her, please? Do you have any question for Susmita Dutta on the future of synthetic biology and microbiome research? Oyabola, do you have any question for her?
No, sir.
Okay. Ajadi, are you ready for us? Ajadi Ganiu. Ajadi.